We'll continue describing the cell movement during amphibian gastrulation. Gastrulation in frog embryos is initiated on the future dorsal side of the embryo, just below the equator in the region of the gray crescent. Here, the cell invaginates to form a slit-like opening, as you can see from the figure, which is called the blastopore. These cells change their shape dramatically. The main body of each cell is displaced toward the inside of the embryo, while cells maintain contact with the outside surface by way of cylindrical neck cells called bottle cells. And you can see these bottle cells in the figure. It is the very first orange cells that have been internalized. These bottle cells will line the archenteron as it forms. So you'll see that the orange cells will start to line the internal as they internalized. Thus, and the invagination of cell initiate the archenteron formation, and you can see from the figure that archenteron is developing while the blastocele is reduced. And you can see from the figure from the outside how the cell converge from the outside to the inside. The gastrulation in frog begins not only at most of the vegetal region, but in the marginal zone as well, which is not seen in the sagittal section, but you can see from the outside view how the cells from the marginal region is migrating. But from the sagittal section, you only see the cells migrating from the dorsal and the ventral hemisphere. When these migrating marginal cells reach the dorsal blastopolar rib, they run inward and traveling along the inner surface of the outer animal hemisphere, as you can see from the figure. Thus, the animal constituting the lip of the blastopore, either dorsally, ventrally, or marginal, are constantly changing because they push inward and another cells are replacing its place. The first cell to compose the dorsal blastoporal lip, as we said before, which is the bottle cells, they lead the other cells to internalize, and they represent the leading edge and form the leading edge of the archenteron. And these cells will form in future the pharyngeal cells of the foregut. As these cells pass into the interior, the dorsal blastoporal lip become composed of cells that involute into the embryo, forming the precaudal plate cells, precursor of head mesoderm. So that means these precaudal plate cells is replacing the involuted cells, which is the prospective pharyngeal cells of the foregut. The next involuting cells that replace the precaudal plate cells is the chorda mesodermal cells, which will form the notochord, which is a transient mesodermal backbone that play an important part in distinguishing the patterning of the nervous system. As you can see from the figure, you have the orange cells which migrate first, and followed by the pink cells, then followed by the red cells, and you can see that the bottle cells directs these cells for the internalization, and you can see that the blue layer of cells, which represent the presumptive ectoderm, trying to make an epipoly and try to covering the embryo from outside. If we elaborate more on the regions of the gastra, you can see that the blue will form the prospective epiderms, while the orange will form the prospective mesoderm, and these type of cells are have the gene cerebrus is expressed, while dark orange will form the prospective head mesoderm and characterized by the expression of the gene GU2, while the red part, which will give the presumptive chorda mesoderm, characterized by zebra gene expression. At the beginning of gastrulation, the involuting marginal zone IMZ forms, which is composed of chorda mesoderm and the head mesoderm. The vegetal rotation, as you can see from the arrow on the figure, the vegetal rotation pushes the prospective pharyngeal endoderm to the side of the blastocele. You can see in C and D that the vegetal endoderm movement push the pharyngeal endoderm, which is represented by the orange color, forward, driving the mesoderm passively into the embryo and toward the animal pole. The ectoderm as well begins to start epipoly, pushing and trying to covering the embryo from the outside. So in this figure, you can see that most of the endoderm have been internalized. The ectoderm, represented by the blue color, is almost covering the whole embryo, and the archenton is well developed, which will form in future the gut, and the blastocele is highly reduced. And as the new cells enter the embryo, the blastocele as well displaced to the side opposite to the dorsal blastopore. Meanwhile, the blastopore lip expand laterally and ventrally, and we can see this from the figure from the outside. Initially, it is the dorsal blastoporal lip, then start to migrate laterally to form a crescent shape that extend laterally and ventrally until merging to form a complete blastopore. And through this blastopore, you can see the endoderm, which now is termed a yolk plug. And one of the main characteristic or feature of completion or the end or termination of gastrula is the reduction of the size of the yolk plug. With the formation of the ventral lip, the blastopore has formed a ring around the large endodermal cells that remain exposed to the vegetal hemisphere. And you can see from the figure, it shows how the movement occur. So you can see that the arrows, when it's solid, it represents the movement from outside, while the arrows which is dashed represented the movement inward. So the remaining patch of endoderm now is called, which appears yellow in this figure, appears, we call it, it's called yolk plug, and will eventually be internalized. You can see here that gastrulation almost have been done in the first figure, and from the sagittal section and dorsal view of the embryo, you can see that another process is starting to occur, which is the neurulation. It starts with the formation of a neural plate and the neural fold, and it is completed by the formation of a neural tube. So once the gastrulation finishes, at that point all the endodermal precursors have been brought into inside of the embryo. The ectoderm has enriched the encircled the surface, and the mesoderm has been brought between them. And you can see here the developmental stages of the cleavage followed by gastrulation, looking to the embryo from the outside, how the cleavage occurs followed by the formation of blastopore, dorsal blastoporal lip, and the crescent form, and then the migration of the dorsal blastopore to form yolk plug, and then starting of the neurulation and formation of a neural tube. In this figure, you can see the different developmental stages during neurulation: early neurula, mid-neurula, and late neurula from the dorsal side. And you can see how you can distinguish between these three stages by the stages of the neural plate. In the early neural plate is flat, and then the neural fold in mid-neurula start to migrate to the dorsal side until the complete fusing of neural fold to form the neural tube. In the mid-sagittal section, you can see how the neural tube is formed in the mid-neurula, being lined by two layers of the dark blue layer of cells, and in between is the neural tube. And in the late neurula, you can see that the neural tube is covered by the ectoderm, which is light blue. In this section, you can see that in this figure, you can see the transverse section and how the neural plate have migrated with its neural fold. The neural fold migrated dorsally and until complete closure of the neural tube and the complete covering of the neural tube by the ectoderm, the light blue. And you can see that as well that the mesoderm have been well developed between the ectoderm and the endoderm, and the starting of the cavity of the gut have been formed. In this slide, you can see the difference between autonomous and the conditional development in the gastrula. You can see in the first experiment, the tissue is transformed between two early gastrulae. They took the region of cells which will give the presumptive neural ectoderm, and they implanted these cells into a region which will give a presumptive epidermis. The embryo generated only one embryo with one neural plate, because the region that they have taken will initially it will presumptively give a neural ectoderm, but when they are transferred to another region, they decided to make an epidermis according to the location, while they repeated the same experiment, but in this case the only difference, they used a late gastrula, not early gastrula, and they do the same setup, but the embryo developed with two neural plates. That means that the cells that have been implanted have been developed into two neural plates and promoted another neural tube formation. So you can see here what is the difference between autonomous and the conditional development. In another set of experiments, they transferred a region that will, the dorsal blastoporal lip tissue from an early gastrula to another early gastrula and transplanted this in the region that will normally becomes a ventral epidermis. But in this case, they found that the region which have the transferred, the implanted tissue, they develop a second archentron, and the second embryonic axis is formed with a secondary neural tube. That means it took the memory of the location of it and induced a secondary structure. So the embryo that will arise eventually, a second embryo will arise with a jointed embryo. And you can see at figure D, it's a live twin Xenopus larva generated by transplanting the dorsal blastoporal lip into the ventral region of an early gastrula. In this slide, we'll see the amphibian mesodermal differentiation. You can see early after the formation, after the nucleation process, the mesoderm is generally divided into two regions, the somatic mesoderm and the lateral plate mesoderm. Soon after that, the lateral plate mesoderm will be differentiated into two regions, an outer somatic layer, which is near the ectoderm, and an inner splanchnic layer, which is near the endoderm. There is a cavity in between them that is called the splanchocele. Soon after that, the somatic mesoderm will be differentiated into three main regions: the dermatome, which is near the ectoderm, outer dermatome, and an inner sclerotome, which is near the notochord, and in between the myotome, which will develop into muscles. There is a cavity in between between these three structures is called the myocele. We can see here how the mesoderm have been developed with a cavity inside the mesoderm, which is called initially the splanchocele, and then will give rise to the coelom, which will give rise to the peritoneal cavity, which will presumably surround the organ. Another structure appeared well defined, which is the neural crest, which is actually the neural fold have been, the tips of the neural fold have been separated and then got located between the neural tube and on the dorsal side between the neural tube and the ectoderm. And this neural crest will be important in the formation of a lot of structure later on. This slide summarizes the developmental stages of the amphibian development, starting from the fertilization until forming of the limb bud embryo, which is near hatching. So you can see how the cleavage proceed, followed by the mid-cleavage state, late cleavage state, then the formation of the dorsal blastoporal lip at stage 10, then the mid-gastrula, late gastrula, and the neural blade formation starting from stage 13 until the complete closure of the neural tube ended by stage 16, then formation of a tail bud, muscular responses, and then the heart beating starts, stage 19, and then the gill starts formed at the stage 20, and the operculum is formed at stage 24, and finally the limb bud stage 37. Hi, today we'll talk about the embryology of the chick embryo and the embryogenesis of a telomere egg. You can see normally how we can color these eggs like in Easter holiday, and sometimes you can see two yolk inside one when you fry egg in one egg. You can see the developmental stages of the chick embryo live in situ on the top of the egg, and as well you can see in the bottom infograph the different developmental sketch of the different developmental stages that occur inside the egg. Here as well you can see how scientists can break a hole inside of onto the shell, and they can watch embryo from this hole to see the developmental stages of the chick embryo in situ. Here you can see the structure of the typical egg of a chick. You can see there is a concentric circles or layers of yellow and white yolk around the structure in the middle called latebra. This yolk is covered by a multiple layer of albumin, two thin layers, one inner and one outer layer of albumin, separating in the middle between the two layers of albumin there is a fibrous layer. These albumin become twisted at the sides of the yolk to form a structure called chelaza. After that, this albumin layer is totally covered by another layer, which is a shell layer for protection. And you can see how the shell layer is separated at some point with the albumin leaving an air space. Looking more similar to the structure of the egg, you can see the yolk and the cytoplasmic part is shifted to the periphery due to the huge amount of yolk, and we term it here blastodisc since it's on the top of the yolk. And you can see the chelaza and the albumin layers, the egg white. If you look closer into the layers that surround the egg, you can see that there is inner shell membrane layer and the outer shell membrane layer, and covering by a more solid layer, which is the shell and the cuticle, and all of this for protection. Here you can see what kind of cleavage occur in birds or chick. Actually, the egg is either called a telolithecal or a megalithecal egg due to the huge amount of yolk it contains. So if we section the yolk material, we can see that the embryonic disc, and this is actually the region where the embryo develop, is located on the top of the yolk. So we call it embryonic disc or the germinal disc. And this part only which is affected by the cleavage due to the bulky amount of yolk that remained uncleaved. Soon after fertilization, cleavage start. The first one is vertical, and the second one as well is vertical, but at right angle to the first one. For the third one, it moves across the second one and parallel to the first cleavage. For the fourth cleavage, it's an irregular circle cutting the blastomeres into 8 central delimited blastomeres and 8 peripheral continuous ones. After the fourth succession of cleavage becomes all of them are irregular. Soon after cleavage, and this shows the major difference between the three types of blastula, the blastula of an animal called Amphioxus, which is egg is isolocystal, very few amount of yolk, and the blastula of the frog, which the type of egg is mesolocystal, shifted to the vegetal hemisphere, but the cleavage occur in the bottom cells, the vegetal cells. You can see the main difference is in the formation of the blastocele, which in the case of Amphioxus, the blastocele is all over the large cavity, while in the frog it is shifted to the side. In case of the chick embryo, you can see since all the cleavage occur in the germinal disc, which is shifted to the side of the yolk, you can see that the blastocele is limited to this region as well on top of the yolk. And now the cells on top of this blastocele are called blastoderm. So now you'll have the egg looking from above is divided into three regions: the area biliosida and area opaca and subgerminal cavity. The area biliosida actually is one or two cell thickness in center of blastoderm, while the area opaca is a peripheral ring of blastoderm cells that contains cells resting directly on the yolk. While the subgerminal cavity actually it's a fluid-filled space between the blastodisc and yolk. You can see it from this figure, it's a section in the yolk and the top blastoderm layer. To the aim to the formation of a blastocele cavity, which will be circled around with blastomeres, so cells will delaminate, just fall down from the blastoderm to form the hypoplast. So eventually, due to the delamination of the cell, and it will arrange in one layer, So a hypoplast is formed, and eventually two layers will be formed, an upper epiplast layer and the lower hypoplast layer. These hypoplasts are formed actually by the delamination from cells of the epiplast layer. Epiplast is an epithelial sheet that will future form the three-gem layer, and now the blastocele is formed between the epiplast and the hypoplast. You can see in this figure as well that the posterior marginal zone is very evident, from which this posterior marginal zone will project some cells internally and merge with the delaminating cells, and from the cells migrating from the deep cells of the posterior region to form the hypoplast. You can see here as well cells from the entire epiplast converge to the posterior midline. So you can see here, and you can differentiate as well from the top figure, the difference between the area opaca, which means that these cells sits on yolk, and the area polyacida, that means that these cells stay or sits on the subgerminal cavity, because still there is no blastocele have been formed, still there is no hypoplast have been formed. So you can see here the cells all from the epiplast converge and migrate to the posterior midline. This leads to a thickness of the cells that raise a little bit above the surface of the cells, which we call it a primitive streak. It starts to take shape. This primitive streak or this thickening of cells will start to migrate to the anterior, making a thickness of cells that soon will give rise to two ridges on the sides and the groove in the middle, which we'll name primitive groove. So subsequently, the cells move anteriorly to form the primitive streak, which you collectively call the primitive streak. The origin of the primitive streaks mark the posterior of the embryo, so it's originate from the posterior and migrate to the anterior. The opposite end marks the anterior. The dorsal side is where the cells migrate inward. The groove marks the right and the left, so the primitive groove is actually the right and the left. So what's Hensen node? Hensen nodes, it forms at the anterior tip of the primitive streak, and cells involute and ingress through the primitive groove into the blastocele to form the mesoderm. So actually the primitive grooves in the chick embryo corresponds to the blastoporal lips in the amphibian's embryo. So let's talk about the avian blastulation and avian gastrulation. So in avian blastulation, there is two layers, actually upper epiplast and the lower hypoplast form. This hypoplast forms by delamination of cells from the epiplast. The epiplast actually is three epithelial sheets that will form the three germ layers. The blastocele actually the narrow space between both these layers, epiplast and hypoplast. While during gastrulation, cells from the entire epiplast converge to the posterior midline, as we have told before, forming the primitive streak. Subsequently, these cells move anteriorly to form a ridge, the primitive streak. Hensen node forms at the anterior tip of the primitive streak, and cells involute and ingress through the primitive groove into the blastocele to form the mesoderm. The gastrulation is complete in the anterior portion and continues to progress in a posterior movement as the Hensen node regresses. So you can see here the arrows show the movement, and actually you can see a section where you can see that cells, the red cells, have ingressed through the ridges of the primitive streak. They ingress inward. These migratory cells will form the mesoderm, and you can see that cells of the epiplast and cells of the hypoplast will, is surrounding the blastocele cavity. So the primitive streak arises by migrating of cells of the epiplast that represent epipoly. Cells streams toward the primitive streak and then turn under to spread out as a sheet between epiplast and hypoplast, forming a mesoderm. Some involuting cells displaces hypoplast and form endoderm, and you can see from the figure some of the yellow cells just displaced the hypoplast and got located between the hypoplast and your future form the endoderm. Again, you can see here, it's another diagrammatic sketch showing the difference between the area pellucida and area obliqua. You can differentiate between them because area obliqua cells are sitting on top of yolk, while area pellucida you have a cavity beneath the area. That's why it's a pellucida, because pellucida means clear. You can see as well in the bottom figure the migration of the cells all over the primitive ridges of the streak, and you can see they can migrate inward. These migrating cells give rise to two types of cells: a mesodermal mesodermal cells that lodge itself between the epiplast and hypoplast, and the endodermal cells that displaces some of the hypoplast to form endodermal layer. So the primitive streak is characteristic for avian, reptilian, and mammalian gastrulation. These streaks arise from local thickening at of the epiplast at the posterior edge, as we said before, of the area pellucida. And this region is called color sickles, KS, and the epiplast and the epiplast above it. What's the check organizers? You can see from this simple figure the relation between the area obliqua and area pellucida, where is color cells locate, the marginal zone, and in the middle there is of course on top the epiplast, below it is a hypoplast separated by the blastocele. So the organizer of the chick embryo, Henson's node, Henson's node forms from cells located just anterior to the posterior marginal zone. This KS is a local thickening of cells at the posterior edge of the upper layer of the area pellucida, the epiplast. It induces the formation of primitive streak, PS, and Henson node. So you can see here from the figure, you have an egg with before cleavage. Once there starts the cleavage and finishes, you can see that the red region represented the area pellucida, while the green region represented the area obliqua. You can see that color cells starts at the posterior marginal zone of cells from thickening of the cells. The epiplast and the middle layer in the anterior portion of the KS becomes the Henson node. This posterior portion of KS contribute to the posterior portion of the primitive streak. You can see here from a side figure, you can see if you look at the sidewise, you can see where is the marginal zone located with respect to the area obliqua. And you can see that the posterior marginal zone anterior to the posterior marginal zone, you can see the thickening of cells represented by color sickles, and you can see the relation of the epiplast and hypoplast. Actually, this figure trying to pinpoint where a color sickle exists. The Henson node has long been known to be the avian equivalent of the dorsal blastosporal lip. It is the region whose cells become the pre-chordal plate and chorda mesoderm. It is the region whose cells can both induce and pattern a second embryonic axis when transplanted into other location of the gastra of the chick embryo. It's the same like the experiment which I have done in the previous lecture when they moved the dorsal blastosporal region into another embryo, and it developed into an embryo with a secondary axis and the second neural tube. So actually, this it is stressed here that the Henson node is parallel to the dorsal blastosporal lip. This Henson node, it expresses the same marker at the marker genes as the Spearman organizers in amphibians. Moreover, the Henson node can induce neural tissue when grafted into fish, amphibia, or mammalian embryo. And you can see here when they transplanted the area of this Henson node or color sickle, they transplanted from the posterior marginal zone, they transplanted to another region in another embryo. They found that another primitive streak is induced to form. In another experiment, they transplanted the Henson node from a duck embryo into a region in the chick embryo. It's not the corresponding region. It's just a region in the epiplast, another region. These transplanted Henson nodes induce the formation of another neural tube. beside the host embryo. So you can induce a new embryo by transplantation of Hensen node. Hensen node from the duck embryo is transplanted to the apiplast of chick embryo. So as you see from the third figure, that a secondary embryo is induced, as evident because there is a neural tube have been formed. Looking more deeply into the formation of the blastoderm, you can see this like a side representation of what is happening, and there is a color coding, and this is from the other one from the ventral side, what you can see, and this is from the dorsal side. So you can see here that it starts with epiplast. You can see that the hypoplasts start as islands because they are delaminating from the epiplast, and you have the posterior marginal zone with the collar sickle, which is the thickening of cells. So hypoplast island can be seen as well as aggregation of hypoplast around around KS. You can see here that hypoplasts start to extend, trying to forming a layer and got connected to the posterior marginal zone. So just prior to primitive streak formation, the hypoplast island have merged to form the primary hypoplast layer, which meets the endoplast cells and the primitive cells at the KS. You can see this hypoplast represented in the on the right figures by the dots in green. Initially it was like island, then start to form from the posterior a sheet of cells that migrate to form a complete sheet at the interior. Finally, you can see that it got connected with the primary hypoplast cell, the green ones, got connected with the blue ones, which is the secondary hypoplast cells coming from the posterior marginal zone. And then the secondary it starts to migrate anteriorly to form a complete layer. After that, it starts the primitive streak in the middle layer, represented by the blue color here, sorry, by the red color here, starts by stage 2, 6 to 7 hours after the egg is laid. The primitive streak cells, which we said before is like a local thickening of cells, forms a third layer that lies between the hypoplast and the epiplast. By stage 3, up to 13-hour post-laying, the primitive streak has become a definitive region of the epiplast, with cells migrating through it to become the mesoderm and the endoderm. And you can notice from the figure that yellow cells and red cells start to migrate inward through the primitive streak and got internalized and lodged themselves between other cells. The mesoderm between the epiplast and hypoplast, and the endoderm lodge itself inside the hypoplast cells. So if we look closely into the migration of endodermal and mesodermal cells through the PS, the primitive streak, you can see here they labeled the cells which are migrating, and you can see here the direction of movement starting first from the primitive streak that migrate from the posterior to the anterior, and then cells from this primitive streak, which appears labeled by the yellow dye or yellow fluorescence. You can see that it migrates from exterior to interior, and you can see how, by looking to the arrows, how the migration occur. Again, it's another figure showing the arrows, the movement, which is color coded, how the cell migrates rolling over the primitive streak. The red cells is the mesodermal, and the yellow cells is the endodermal. In this slide, we summarize the developmental stage of the chick embryo. You can see that the start of thickening at the posterior edge of the area biliosida, which is called the blastoderm, the posterior region of the blastoderm. Soon after that, this thickening start to migrate to the anterior, forming the primitive streak, which is taking shape while migrating to the anterior. Soon after that, the primitive streak reaches the anterior, with the head of the primitive streak is called Hensen node. These three stages show the dorsal view and formation and the elongation of primitive streak. In figure D, you see that the head process started to develop with the regression of the Hansen node and the primitive streak. Soon after that is further development of the head region through the formation of the ectoderm of the head fold and the anterior border of the mesoderm and the neural fold and the notochord, and starts to appearing the somite on the sides of the neural tube formation, and the primitive streak is more regressed to the posterior. At the end, you can see that the neural folds start to be more clear, and the somites start to be segmented, and the endodermal has now ingressed beneath the epiplast, and the primitive streak is markedly reduced. And these three figures show the formation of notochord, mesodermal somites, and BS regression, primitive streak regression. If you look again to a diagrammatic figure, you can see that where is the Hansen node located at the beginning of before the formation of the head fold, and soon after the formation of the head fold and the somites start to appear on the sides of the neural tube, the primitive streak start to regress to the posterior with the Hansen node until the somite starts to appear and the closure of the neural tube. So actually, if we'll talk about the Chick fate map, it's very hard to recognize the fate map contradicting to the amphibians, which we can recognize the fate map even on the egg itself. But since the chick embryo, the blastoderm cannot be mapped, as the early chick embryo is small and there is much more cell proliferating and extensive cell and tissue movement, the early fates are not known. Later, when the primitive streak forms, the three-germ layer can be mapped as seen from the figure. So once the primitive streak formed, you can see that the Hansen node will give presumptively the notochord, will direct the formation presumptively of the notochord and somites. And you can see in the gray region, which will give the extra embryonic mesoderm, and the orange region will give the somites and lateral plate, while of course the dark blue will give the neural ectoderm and the light blue will give the ectoderm. Here you can see the neural tube formation in the chick embryo. It starts, of course, with neural plates. On the side, there is a neural crest, which is bounded by the epiderms, and soon a groove is formed, induced by the presence of the notochord beneath it. And this groove, this folding, occur at the medial hinge region. Soon after that, the folding starts, and the ridge starts with the elevation of the neural crest. Soon after that, these convergence of the neural groove trying to close the neural tube and form a complete neural tube, the conversion of the neural folds occurs as the dorsal lateral hinge points. Cells become wedge shape, and the epidermal cells push toward the central. Soon after that, the neural crest, which is the ridges of the neural fold, which link the neural folds with, which link the neural blades with the ectoderm. Soon after that, the neural crest cells link the neural tube with the epidermis, as seen from the figure. The closure happens, and the neural crest then disperse, leaving the neural tube separate from the epidermis. So, what is the neural crest? This slide shows the ectodermal derivatives in the chick embryo. The ectodermal can, from the ectoderm, the neural tube arise, neural crest, and the surface ectoderm or the epidermis. From the neural tube, you can see the brain, neural pituitary, and the spinal cord, and motor neurons and retina are differentiated. From the neural crest, a lot of structure as seen is differentiated, including the dentine of these facial cartilage, melanocytes, adrenal medulla, and the peripheral nervous system, including Schwann cells, neuroglial cells, sympathetic nervous system, and the parasympathetic nervous system. While from the surface ectoderm, the epidermis, a lot of structure appear, like the epidermis, hair, nails, sebaceous gland, olfactory epithelium, mouse epithelium, including the anterior pituitary, tooth enamel, and cheek epithelium, and lens and cornea. In this slide, you can see the 24-hour chick embryo, and you can see the cephalic head region has undergone the neurulation, while the caudal region are still undergoing gastrulation. And this is because actually the neurulation starts early in the anterior part with the regression of the posterior groove. So you can see that it is highly developed anteriorly more than posteriorly. And you can see the location of each germ layers: ectoderm, endoderm, and mesoderm. The mesodermal development in frog and chick embryo, actually it's both of them, frog and chick embryo, follow the same pattern. So you can see the chick embryo have the neural tube in the middle covered by the ectoderm. On the sides of the neural tube there is the mesodermal somites, which will give to the lateral mesodermal plate. This all on top of the endoderm, showing the primitive gut. You can see as well the notochord is lying just beneath the neural tube. So hypothetically, if you do these cuts on the sides of this germinal disc and you roll these sides in the form of the tube, you will see a very similar structure to the one we have discussed in the frog embryo, with all the same pattern. So basically both of them share the same pattern of formation of differentiation of mesoderm.